Hi guys, sorry I'm not there. Evan's sick again. Um, Hopefully you are working with a partner right now. One of you should have the video up on your iPad and be using Edpuzzle to access that. And the other one should have open their note sheet from Friday that we were working on. On Friday, we got up to here where we figured out what these amino acids coded for. We skipped this part about anticodons, and we're going to skip it again for a few minutes. We will come back to it. But I want you guys to focus first on the translation process. And that's what we're going to go through briefly here. Feel free to pause the video at any point in time to make sure that you have the notes down that you need in terms of each of these different steps. We talked a little bit about that translation process on Friday in terms of, okay, if you're given DNA, you know how to code it into mRNA, and then the translation process is how do we read that mRNA and turn it into a protein? And that's what you guys did for homework over the weekend. You took that mRNA strand and you used your codon chart. If you remember, your codon chart is this awesome wheel here, and you used that to figure out which amino acids there were. Those amino acids built the protein. What we want to talk about here is what that translation process actually looks like within the cell, right? So you should be on this part of your notes, at least one of the partners, and you should be writing down the notes in your note sheet. When you're done with it today, the partner who's working in the note sheet needs to email the notes to the partner who is working in Edpuzzle so that both of you have a copy of the notes. All right, let's get started here. Okay, as a quick refresher, remember our main goal in translation is to take mRNA and make a protein. Okay, this, what we're talking about today, is how it happens in a cell. Step one is pretty simple. The mRNA molecule leaves the nucleus. We talked about that. That was the very last part of transcription. And it's going to bind with the ribosome. When it binds with the ribosome, it's going to start at that start codon. So in this first picture, you really want to make sure you know that this gray thing, this is the ribosome. And a ribosome actually comes in two parts, the top half and the bottom half. And the mRNA is going to be this. That's going to be read by the ribosome. All right, so as I said, step one, the mRNA molecule has left the nucleus and binds with the ribosome. It's always going to begin at that start codon of AUG. If you need to write this down and need more time, hit pause. Step two is where the ribosome starts to read that mRNA molecule. We're going to see another player come into play here. That additional player is going to be our tRNA. If you remember from Friday, we said that the purpose of tRNA is to transfer an amino acid over to the ribosome. And it has attached to it an amino acid, that's this red part, and there's three very special nucleotides that are at the bottom, okay? Right now, I want you to label this an anticodon. We're going to define this a little bit more in a little bit. I just don't want us to get too confused with it. So a codon is on mRNA, and it's the 3 letters that are being read. An anticodon is a part of tRNA, and it's the 3 letters that match up. So if my codon, in this case, was AUG, my anticodon is the complement of that, or the UAC. Okay? So what's happening in this step in the cell is beginning with that first codon, the tRNA molecule, that's this blue one, ends up bringing an amino acid over that matches up. Okay? And it knows to stop there because it has that anticodon that can bind to it, because they are those complements. We like to match the opposites together. So the ribosome is holding the mRNA in place, and we're going to see that tRNA molecules, the ribosome binds the tRNA molecules with mRNA according to codon-anticodon pairs. It's kind of the big idea. It holds it in place for a second as well. That's step 2, this idea that we're binding this tRNA temporarily to the mRNA based on those opposite base pairs and bringing the amino acid over. Pause it again if you need to. In step 3, let's label all our parts here so we know what's going on. The blue is our tRNA, and we should see a second tRNA molecule over here. Okay, we should see 1, 2 anticodons. We should have our mRNA down here. And I should see, here's my first codon, here's my second codon. And these red bits are the amino acids. What's occurring is the next tRNA molecule comes in, binds according to those codon-anticodon base pair rules. So again, if we're looking very carefully at my second codon, it was UGC was my second codon, the UCG. And then my anticodon, the A paired with the U, the G paired with the C, and the C paired with the G. And it brings that next amino acid over. What's great about this is because the other amino acid is still hanging out there, the ribosome was holding on to it, we can bind these 2 amino acids together. And once we start to make a chain of amino acids, that's going to begin the beginnings of our protein. Okay? Pause it here if you need to write something else down about what this step is. The fact that the tRNA brings the next amino acid over and they get bound together. In step 4, what's going to happen is now that that first amino acid got bound on, again, if we want to label parts that might make this easier, we've got our red amino acids here. Okay, our blue stuff, hopefully we're seeing that that's our tRNA. This is a tRNA. We have our anticodon, as well as anticodons over here and over here. This would be my mRNA still. And we would have had codon 1, codon 2, codon 3. This would be our fourth codon there. These are my codons. Okay, so as I was saying, in step 4, the first tRNA gets released because its amino acid is already attached. The tRNA doesn't have any other purpose. And everything gets moved down so that the next codon can get read. And this process is just going to keep continuing. New tRNAs are going to come in, match up with the next codon, and the previous tRNAs are going to get kicked out so that there's room in the ribosome to hold on to them. What we're going to find out is this growing amino acid chain is going to be our protein. Okay? I like to think the tRNA is kind of an amino acid bus. It goes, it picks up the amino acid, it brings it over to the mRNA, it knows where to drop it off based on the codon, and it drops it there and leaves it there so that it can bind to the next amino acids. Okay? This keeps going and going and going. I mean, proteins can be thousands of amino acids long until that stop codon gets reached, and then everything gets released out of the ribosome. Pause it if you need to after step 4. That, guys, is the basic process. Okay? This here is a picture of everything that's happening. We're going to walk through this a little bit together. So if you go on your note sheet actually looks like this. Okay, and there's going to be some things that we want to label within it. First, we want to label where each step is occurring, and then we want to make sure we know what all our major players are within this. Okay, so step 1 is going to be over here. This is where this all begins. We're talking big idea here. Step 1 was transcription. And remember, transcription was going to be DNA to RNA. Okay? So if we look here, this purple stuff is our DNA. This here is the DNA that is growing. We are inside of the nucleus. Okay? And this new strand, this new strand here, this is going to be your mRNA. It's what's getting made as we read the DNA. Okay? So step 1's happening inside the nucleus. The major players are the DNA, which is really that big purple molecule. They've zoomed in a little to show us the different nucleotides within that DNA molecule, but it unwound, got unzipped, and we started to create the mRNA based off of those base pairing rules. Then the mRNA is going to leave the nucleus. Okay, and it's headed off to our friend, the ribosome. Okay, so the mRNA can be really long. This whole thing here represents that mRNA. And this is going to be where we get into step 2. Step 2 is translation. And as we've been talking about, translation is the process of going from RNA to a protein. Okay? We are going to be in the cytoplasm, hanging out at the ribosome for this whole process. So you could label all of this over here as our cytoplasm. Okay, If we want to label things down in here, this is a zoomed in picture of translation. This is my mRNA, this strand down here. A 3 set of bases is going to be our codon if it's on the mRNA. The green things here are my tRNA, and attached to each tRNA, we need an amino acid. Then, as the amino acids bind together, we're going to get a growing protein. Okay? And that growing protein will eventually become the protein that we were trying to build. The one thing we don't have labeled on here is that anticodon. The anticodon are the 3 bases that are found on the tRNA. Like I said, we didn't really go through that too much. Sometimes anticodons can confuse us more than they can help us because people get confused about what to use on the chart. You guys have been using the chart the entire weekend. You didn't know about an anticodon because the chart, we always use the codons. We always use the mRNA. The anticodon is just kind of a little extra. It's a way for the tRNA to know where it fits in. Okay? So if you want to go back to this part of your note sheet, we should probably add just some details for ourselves so that we know what we need to about these anticodons. So get to that part of your note sheet. I'm going to scroll back up to where we've talked about it, or where we should have talked about it, and then we'll get going from there. Where did I put it, guys? I was smart and I put it where we needed it. Sorry that you have to watch all of that moving. An anticodon, a couple of things that are really important. It complements the codons, meaning it follows the base pairing rules. A goes with U, G goes with C. It's found on tRNA. That's huge. Okay, and it matches up to the codons, which allows it to bring over that proper amino acid. This is one of the most important statements of this entire slide. Do not use anticodons with the chart. The chart is only for codons. Okay? So look at the example and take a look at your chart. In this example, we know the codon is AUG, which means our anticodon would be UAC. The A of the codon matches with U. The U of the codon matches with A. The G of the codon matches with C. Now, using that information on your chart, you should be finding that the amino acid is methionine. If you got something else, it's very possible you were trying to use the anticodon. But we always, 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 always use the codon with the chart. All right, now that we've kind of gone through some of these notes, the last thing I want you guys to do today is go through and try to fill out these power notes. Now, the power notes that are found on Schoology already have all of these words written in and the color-coded process. I just want to talk about this so you make sure you know what you're doing with it. When it asks for the reading frame, you're just going to answer this simple question. How many bases are read at a time? Okay? So do I read 1 base at a time, 2 bases, 3 bases, 5 bases, 10 bases? How many bases pair up with an amino acid? The common language, all I want you to know is that the common language we're using is this idea of the genetic code. You don't have to write anything else in that box. You should describe what a codon is. Where is it found? What type of molecule? How many bases long is it? When do I need to use it? You should know what the start and stop codons are. We talked about those on Friday. They're in your notes. For the ribosome, there's 2 bullet points. 1, I want to know what the purpose of a ribosome is and what makes up a ribosome. We talked about that briefly. Anticodon, you're just going to describe it. Where is it found? What is its purpose? What happens? And then for tRNA, what's its main purpose? Okay, so that's the top part. Then down here in the bottom part, you're going to describe the process of translation in a cell. So this, I know we talked about 4 steps and this only has 3. This should be step 1 and 2 from your notes that we just took. Remember, if you were the person taking the notes while this video is playing, you need to send that to your partner. Step 2 of this process would really be our step 3, and step 3 would be the step 4. So just combine step 1 and 2 because step 1 was pretty simple and basic. Then over here, in this area, I would like you to label the parts, and I've tried to color code them for you. Part number 1 is pointing to the green circles. Part number 2 is pointing to this whole thing, both green things, what's happening there. Part 4 is pointing to this entire thing. Okay, the purple with the little green thing attached. Part 3 is pointing to the orange blob. Part 8 is focused on those 3 bases right there. Part 7 is focused on this entire strip. Part 6 is also focused on the orange blob. And part 5 is focused on these 3 bases here. We will go over the power notes for sure tomorrow in class, as well as the homework from the weekend. If you have any questions, please send me an email. I will try to be watching it, try to be helping. Mr. Winter is also a great resource. Please feel free to ask him some questions as well. Thanks, guys. Have a great time. Sorry we did notes this way, but I think you can handle it. And we will get back on track tomorrow in class.